We are just going to wait a few minutes until everybody gets into the room. We're just giving it a couple of minutes till people get into the room. Good evening and welcome to tonight's STEM Cafe. What lurks beneath, how soil microbes support plant health. Our STEM Cafes are brought to you by NIU STEAM and the Center for P20 Engagement at Northern Illinois University. I'm Dr. Judy Diamond, organizer of the STEM Cafes for the past 8 years and your host for this evening. NIU STEAM refers to science, technology, engineering, the arts, and math. STEM Cafes are fun, casual gatherings where adults can eat, drink, and chat with STEM professionals about hot topics and the latest research. We usually hold our STEM Cafes in restaurants and look forward to getting back to our restaurants. Tonight you will need to provide your own favorite snacks, drinks, and carryouts. In the meantime, our partner restaurant, Fatty's Pub and Grill in DeKalb, is offering a 10% discount to anyone who mentions our virtual STEM Cafe. For those of you who have not attended our STEM Cafes before, our format for this evening is as follows. Our speaker will talk for approximately 40 minutes. During the talk and following the talk, you can send in your questions and comments. We know that many of you are experts in the audience as well, so often our attendees add to the discussion with their comments and insight. We will end by 8 p.m. If you experience difficulty hearing during this presentation, you can find a volume control in the top left corner of your screen that can be adjusted to your liking. The cafe will be recorded. It will be posted on our website in about a week, as we need to make sure that we have closed captions and this is accessible to all. Our speaker tonight is Dr. Wesley Swingley. He has been a speaker for us before on astrobiology. Dr. Swingley, Associate Professor in Biological Sciences at NIU, describes himself as an astrobiologist, a biochemist, a microbiologist, and conservationist. He grew up in a tiny Montana town, but has since followed school and work all over the world. He began his science career with a heavy interest in astrobiology and the evolutionary adaptations that allow life to inhabit any and all environments on our planet. He describes himself as a space nerd who is always looking to link theories of microbial ecology to life we might find in our solar system or beyond. Tonight, Dr. Swingley will discuss how microbes in soil affect plant health and how plant health, in turn, affects humans and the planet. He will share insights into the pivotal roles microbes play in shaping ecosystems. Dr. Swingley has a National Science Foundation grant to conduct a study at the Morton Arboretum and the Natchusha grasslands on how bacteria and fungi interact with prairie plant roots. His team is gathering plant traits, secretions from roots of living plants, and soil communities. These data will be combined and analyzed using new machine learning approaches to build predictive models. He is working in partnership with researchers at San Diego State University, Middle Tennessee State University, Lawrence Berkeley National Laboratory, and the Morton Arboretum. Dr. Swingley, would you like to begin?
Thank you, Judy. Can everybody hear me okay? You can hear me? Judy?
A little louder.
All right. Well, hopefully it should be good for everybody. Thanks again for having me to one of these STEM cafes. As Judy mentioned, normally these things are a lot more fun in terms of drinks and hanging out with people, but hopefully you have good people there with you that you can share a drink with and have some fun. Today I'm going to be talking a bit about some of the things that Judy mentioned. I'll be talking a lot about soil microbes and their interactions with plants, but a lot of what I'm going to be talking about is kind of the difficulty in studying some of these big issues and some of the big questions that we want to know about plant health and environmental health and all of that. But one of the first things that I always have to do, I'm a microbiologist, and of course microbiology has that word micro in it. And so one of the things I talk about a lot is just what does that mean? Oh wait, actually first, sorry, I always put this slide at the end, and I know that I should put it at the beginning. The most important thing in anything that I do are the people that I work with. And I've been very fortunate to work with some amazing people. Over the years in my lab, I've had a lot of students who have been working on projects that have led up to this project. You'll see a lot of these people in the slides to come. Jared and Renee were two of the first students that I had on a summer fellowship that did a lot of the work that led us into this project. And then, of course, I continue to have students working on this project actively, and even people who are not students. That last person, Rush, is a prospective student, somebody who's just very interested in getting into science. And so this is a project that people are working on at all levels, both students and community members. And I couldn't do any of it without that. The collaborators, as were mentioned, are all over the place, especially for this new project. We have Nick Barber is one of the lead collaborators at San Diego State University, and some of you probably know Nick. He was here at Northern Illinois up until a couple years ago. But the rest of our project team is, I'll talk more about them when I get to them, and they're a really diverse group of people, and it's really amazing to work with them. And then, of course, I can't do a lot of this without Natchusha. The staff at Natchusha have been amazingly helpful to me and other NIU researchers over the years in terms of supporting the scientific questions that are really profoundly important and really can be done in a level of detail at Natchusha that can't be done in other natural systems. And a lot of this work has been funded through Friends of Natchusha, which is a group associated with the Natchusha grasslands. Completely community driven. They've funded a couple projects through my students. And as Judy mentioned, we have a new project coming up that really just kickstarts off of some of the things that I'm going to show you today. But as I hinted at, the first thing I always have to do is kind of convince people that microbes are real, or kind of convince them to think about microbes in the right ways. And so the first slide here really just shows you what your eye can see, right? We have a lot of very big things on our planet. These redwoods, I took this photo a couple of years ago in Northern California. These trees can reach up to 350 feet tall. They're some of the tallest, some of the biggest things on our planet, right? That we see those, we think about our biosphere in terms of these big trees and these big animals and things that are moving around on the surface. We also have a lower limit to what we can see, and this is my little attempt at taking some kind of muddy soil here and picking apart some of these soil particles that go down to about half a millimeter or smaller. And just to put that in perspective too, with your hair, the number that people often throw out for human hair is about a tenth of a millimeter, 0.1 millimeters. So we know that we can see human hairs, but just barely sometimes, right? I didn't use one of my hairs as an example because my hair is super coarse and it'd be bigger than that soil, so that's no fun. But these little soil particles really are still quite huge. And so I'd like to convince you that this big here and the tiny little things in my hand, these are both really big in terms of what we're looking at with microbes. And so that soil particle, really what I typically think of it as, and what I describe to my students when I talk about this, is they're little apartment complexes. That one little teeny piece of soil that you can barely see looks a little something like this, where you have the apartments that are on the surface, right? They get lots of nice air and light, fresh air and light. The dank hallways in the middle where it's humid and there's lots of weird smells. That's all this organic matter in the middle. There are a lot of, there's a wide variety of locations just within this one little soil particle. And it's also massively huge compared to these microbes. So these microbes can inhabit these little environments within that soil particle. And you can have in that piece, maybe even this smallest little piece in my hand here, that can house thousands to millions of microbes in just one little soil particle. And so this is really a big cartoon view of that, but there's a lot of different spaces in there where they can live, and we think of it as kind of a homogeneous thing, but they're very different environments, this outside surface and these little inner surfaces. And those microbes that I'm going to be talking about for the most part today, these bacteria, are very, very small compared to what we can see with the naked eye. And so this figure here is from the textbook that I use for microbiology, and it's a really good look at the upper limit here, this paramecium. A lot of you have probably seen a paramecium or something like that in high school biology class. Those could be seen with the naked eye. They're just barely at that limit of what you can see with your naked eye. But most bacteria are in this range down here with E. coli of about 2 microns, 2 micrometers. And that 2 micrometers is, or 1 micrometer, here, let's do easy math. That 1 micrometer, that's a million times smaller than a meter, right? So we know what a meter looks like. A millimeter is 1,000 times smaller than that. A micrometer is 1,000 times smaller, and it's 100 times smaller than what we can see with our naked eye. So if we can see this paramecium, it's 100 times smaller than that. And to put that into perspective, those redwood trees that I showed at the beginning, those are not even 100 times bigger than me, right? We'll say 6 foot, I'm not 6 foot, but if I'm about 6 foot, those trees are only 300 feet. They're only about 500 or 50 times bigger than me. So that difference between me and those redwood trees is not even as big as this difference between the paramecium and the E. coli. So you can really see that the limit of what we can see from these microbes gets to be very difficult. And then if you talk about things like viruses, they're even smaller. So viruses are off the scale shown on this little figure here. But we know these microbes do a lot for our ecosystem, and even though we can't see these microbes, we know that there's a lot happening through primarily through research and through experience with these microbial systems. And in terms of plants, a lot of that microbial influence is in the roots. So the roots here are really the gut of the tree, right? This is where all the action is happening in terms of the tree interacting with its environment. And so these are one of the biggest sources of nutrient uptake for the plant, nutrient and water uptake. And of course, the above ground is where all of the action is happening in terms of pulling in carbon for building their biomass, that carbon dioxide to build up their biomass, and absorbing the sunlight to power all of that. But these interactions within the roots drive a lot of the nutrient uptake in the roots. And we'll get talking a bit more about this, and I'll zoom in on this figure a little bit. But one of the biggest areas that has been talked about for a long time in terms of plant root health is this one right here, nitrogen, right? Nitrogen fixing bacteria. And I think most people, whether they think of themselves as scientifically minded or not, have probably run across this term nitrogen fixation or heard about nitrogen fixation in roots, and that is always associated with legumes, right? So legumes, soybean being the best example, is a plant that we grow that we know bring their own nitrogen into the roots, and that nitrogen can be a limiting nutrient. And so the fact that these plants can make an association with bacteria to get more of this is really powerful in terms of growing those plants. They're independent from things like fertilizer, or at least they can be independent from fertilizer. Where something like corn or most of the other crops that we grow, we supplement those heavily with nitrogen in order to feed them that nitrogen that is one of their limiting nutrients. And so that's really been one of the biggest focuses in terms of plants, but there's so much else going on, and some of the other major nutrients that life needs we think about pretty commonly, such as carbon, where they're of course getting their carbon from the atmosphere, but also phosphorus, which is another common component of fertilizers. So phosphorus and sulfur and some of these other compounds that the soil may be lacking in ways that bacteria, or humans in the case of farming and cultivation, facilitate and help improve that plant growth, right? And the other thing to keep in mind here is this is a cartoon, but this is fairly representative of many plants where you have this root system that is really as vast as what we see above ground. And this root system can have a huge surface area contacting much of the soil around that plant. And when we talk about the fungal members of this community, they extend that root system even further. But I do want to talk a little bit about the name of this, right? The name of this talk was What Lurks Beneath, and that what lurks beneath is not limited to topsoil. We know now, as of relatively recently, we know that the biosphere of Earth goes down much further than we ever anticipated. And that's through a lot of scientific study looking at mine shafts. You can access things directly in a mine shaft, or doing cores deep in the crust or deep in the ocean crust. And we've found that we see evidence for life miles underneath the surface of Earth. And we can quantify that. And a lot of people are really excited about this and really excited about doing the math on just how much biomass that is, just how much is happening underground, because we live on just this teeny little skin on the top of our planet, right? One of my favorite visuals that is not quite accurate, but is fun to think about anyway, is if you take a basketball and you breathe on that basketball, that breath, that little thin layer of breath that you've put on the basketball is about the thickness of Earth's atmosphere. It's actually much thicker than Earth's atmosphere. So we really live on a very fragile little skin on the surface of our planet. But this biosphere that microbes live in extends for miles beneath the surface of the planet. And the density is not as high. These microbes are not really very active. They're not doing stuff very fast, but there's so many of them that they control a huge portion of the nutrient cycling in our planet. And over the history of our planet, these have probably contributed greatly to all of the nutrients that have moved around our planet and changed our planet over time. So it's a really cool thing. This figure here is near and dear to me because I know a lot of people in this community that work in this area, people who are interested in astrobiology like me and interested in thinking about life on other planets like Mars, are very interested in what's happening underneath our crust, because we expect that there might be the same thing happening beneath the crust on Mars, where life is sheltered from the radiation of the sun and the harsh atmosphere, and maybe in closer contact to water, to liquid water. But back to topsoil. So the one thing I often say, and again, because I think I have a spacey context, is that studying microbes is a lot like studying animals from space. And so I spent a little time finding a Google Maps view of my hometown, since it's Sims Creek right here. As Judy mentioned, I grew up in a hometown of less than 200 people. There are probably more cows in a mile around my town than there are people. And so I zoomed in a little bit here so that we could see these cows. These are a few, I think they're cows. I don't know, they're very blurry, right? So these little things that it took me a little while to dig around and find on the map And then it took me a little while to figure out whether I want them to be cows or sheep. I don't know. That's really a lot of what microbiology looks like, because if you take your normal light microscope that you all have probably seen before and you look at some microbes under that, you see this. You just see little lines or dots or curly things, and you can't really make much of them. You can't tell if these are cows or sheep. Maybe somebody can. I can't. And you can't tell what type of microbes these are. These happen to be E. coli, but if I looked at these under a microscope, I would have no idea what kind of microbes these might be. And that's because you're just looking at the shape. You're looking at this little shape, and I'm here to tell you that most microbes are very boring looking. Most microbes look like this. They're a little rod or a little dot, something like that. And so when you look at them under the scope, you really don't get much. And so for a long time, since the advent of microbiology, which hasn't been very long, just a couple hundred years, people have been doing other types of tests to analyze what these things are doing. And they're always indirect, or they're group analyses where I can't tell what this one microbe is doing. But if I grow a lot of these microbes together, then I might be able to see more. I might be able to see what the whole colony looks like, see what they look like growing together. Or I might be able to see what metabolism they do, what types of sugars do they eat, what types of byproducts do they produce. These are things you can do in a large culture that you might not be able to do on each individual. And this is still a problem. I say we've been doing this for 200 years. We haven't changed a lot. Analyzing microbial metabolism still is mostly done that way, by looking at the whole community. And that is a challenge, because each member of this community is probably not doing the same thing. We know that there are cheaters and there are workers, right? There are some microbes that just take from the rest of them and never actually do anything, just like real life. But this is hard, and this is a really tough problem when we're trying to understand how microbes are interacting with a system, especially when we're thinking about a system that doesn't just have one microbe in it. And when we're talking about plants and soils and things like that, these are wildly diverse. These communities have basically the entire diversity of planet Earth in 1 teaspoon of soil. There was a study that came out a few years ago now, where scientists took soil from different parts of Central Park in New York City. And from those samples, they sequenced the microbial communities, and they found basically the full diversity of microbial life that we know of from the planet, all just in Central Park. All of the microbes, maybe not all, but a representative of most of the microbial groups were found in that sample. And so that can be a big challenge, because we're trying to look at something that is incredibly complex that we often know very little about. And so this is what that usually looks like. And my apologies, I won't make you look at this very long. I've given this slide in a talk in person before, and you can hear people groan when this comes up. But this is really what our understanding of microbial life interacting with plants looks like. And this is from that slide earlier, where we have this interaction between microbes and plant roots. And we have the roots on the left here and the microbes on the right. But everything is very, very squishy in between. Everything is very blurry. And this is an area that I'm really interested in clarifying. And this is a lot of the project that I'm working on, is understanding directly how these microbes are interacting with these nutrients. We'll get off of that slide. That's horrible. But one of the reasons is, one of the things that everybody's very interested in thinking about sustainability and economy and just the human race moving forward, is how can we best leverage what we know about microbes, what we know about science and technology, to improve our plants. The very real problem that almost every one of you probably know, because most of you are in Illinois, I presume, is that we spend a lot of time, a lot of money, and a lot of ecological damage in making our plants grow faster and better. And that's typically through fertilization. So this boy here is the problem, right? Corn is a huge industry in Illinois, but it's not just happening with corn, where we dump huge amounts of fertilizer, mostly in the form of nitrogen fertilizer, ammonia fertilizer, onto crops in order to make them grow better. And we do that because we know it makes them grow better. But much like vitamins, if any of you have taken vitamins, you know that you take your vitamins and then a few hours later you pee and your pee is fluorescent yellow, right? And that's because you're peeing out most of those vitamins. You take a huge quantity of vitamins that your body can't absorb in the hopes that your body will absorb a small portion of that. And that system holds true for a lot of things in terms of fertilizing. We fertilize fields with way more nitrogen than they can take on to maximize the amount that they take up. But that's a big problem. And that is a big problem mostly because of microbes. So if any of you have worked with ammonia, ammonia is not water soluble. Ammonia in our fields around here is usually pumped in as a gas, anhydrous ammonia, meaning it doesn't have water associated with it. And that ammonia very, very rapidly is broken down by microbes into nitrate. It's converted into nitrate. This is a very fast process, very energy efficient process for microbes. And that nitrate generally is not as available to plants. And that nitrate is also very water soluble. So what happens is you put this agricultural management on here, right? You put that fertilizer on there, and that fertilizer is taken up by plants to some extent, but much of it is washed away. And that's washed away from Illinois. Maybe we don't care about it, right? We're at the northern end of the Mississippi, so what do we care if our water goes down to the Mississippi and into the Gulf of Mexico? That's a major concern. It's not just an ecological concern, but it's a social concern because we're passing our problem down the river to somebody else. So people are interested, of course, in looking for ways around that, and not just looking for ways around that from an ecological perspective, but also from a financial perspective. Everybody wants to make a living, and everybody wants to make the most out of the crops that they have, and so things like very basic organic farming or low yield farming are not sustainable in terms of feeding a country, of feeding an industry. And so we need to find ways somewhere in between boutique farming and big industrial farming. And so one hope for that is using microbial inoculants on roots. And so you can take bacteria or fungi and add them to the roots and make those roots grow better. And those roots are, I'll talk a lot more about this later, but those roots themselves are trying to form a microbial community as well. Those roots are putting off chemicals that we call exudates. They exude them, right? They exude these chemicals. And this is a wildly unknown field of biology, what type of chemicals these plants put off. And these chemicals, though, are what attract microbes and what microbes use to metabolize and grow and convert and do all of these things that they do in terms of promoting the plant growth. And this is not necessarily always direct. I think a lot of times when we think about soil interacting with plant roots, we think of these direct connections like those nitrogen fixers, the organisms that work directly with the plant roots to fix nitrogen and help that plant grow. But in most cases, you're having many, many different effects of how those microbes are growing in the soil and affecting the plant roots. And this whole thing here is generally called the rhizosphere. The rhizosphere typically refers to just that area around the root. In a lot of cases, the plant root system is so complex that the whole soil around the root system is effectively rhizosphere. It's effectively in contact with the plant root, but not all of the soil. And even that bulk soil around here that's not in contact with the root will be indirectly affecting it. So in managing microbes, this is actually something that we've been doing for quite a lot of years, but we've been doing it in a very specific way. And that is these sorts of things. These are ones I pulled off of Amazon. I don't expect if any of you are farmers that any of you have used these products, but some of you may have. And I'd love to know if anybody in the room has used any of these products. Maybe afterward you can let me know. But these are fungal inoculants. And so these are all a wide variety here, as you can see, of these fungal inoculants. I like this one because it has extreme spelled with an X, which means it's definitely the best one. And these are inoculants that you put on your plants to get the fungal communities started that can help support those plant roots. These are very, very similar to probiotics for humans. And so if any of you have taken probiotics, I've taken probiotics, they can be useful, but they're also something that isn't regulated very strictly. They're not necessarily backed by a lot of science or a lot of supporting data. And there's been a lot of studies that have looked at the effects of these types of introductions and seen a wide variety of results. So you might have one of these that works incredibly well for corn. You put it on your corn crops and the corn doubles in mass or something. But then if you put that same one on a related grass or on another crop like soybean, for example, you actually can see the yield decrease. So they're not a one size fits all for every plant. And the types of plants that we grow for our food are very phylogenetically diverse. They're very different from one another. So we don't expect that one of these plant supplements can actually, sorry, I'm gesturing with something that's not in my hand. We don't expect that these plant supplements will actually do a lot for every single plant that we try. So you might have to test out different plant supplements to see which one works for your system. And that's a bit of a problem. I am by no means a fungal researcher, but I did want to talk about this because this is a really critical aspect for plants. And so nearly all plants, roughly 90% of plants form these associations with fungi, and they're called mycorrhizal fungi. And so you've seen this word before, this rhizo word. It just means roots. So these mycofungal root associated groups have evolved to benefit both parties. The plant is supporting the fungi by providing nutrients for the fungi. The fungi are extending the root system of the plants in many cases, spreading out through the soil, forming a lot more surface area contact with the soil to bring in more nutrients for the plant. So this is a very well-known, very well-studied symbiotic relationship. And there are a couple major types. The endomycorrhizal fungi, endo meaning inside. These actually work directly with plant cells. Ectomycorrhizal fungi are superficial. And the ectomycorrhizal fungi are a much smaller minority of these types of fungal groups, and they're typically associated with trees. I just stole a little picture of a beech tree here. But interestingly enough, some of those, I flipped through the ingredient list on some of these inoculants, and several of them had these ectomycorrhizal fungi. And that's very intriguing because I don't know that those work for many non-tree, non-woody plants. So very intriguing that we see those there. The endomycorrhizal fungi, I'll pull my arrow back here because this group here really is the king of these endomycorrhizal fungi. So a lot of the supplements you see will have glomus or rhizophagus, and this is kind of a renaming. That's why I have a slash there. So this glomus group, as far as I can tell, and again, this is me not being an expert in this field, this glomus group, it was a very broad genus, and it has since been split off. And some of the major ones, such as one of the strains of rhizophagus, have actually been differentiated, and this is now its own genus that's one of the major plant inoculants. But these they are very broad scope. So this is a really good example of something that we can add to most plants, and most plants will like it, and it may help improve their growth. And then there are a lot more tailored groups of these endomycorrhizal fungi that may support specific types of plants. But what was interesting to me is that a lot of these inoculants, even if the package itself, the package 100% on the front said fungal inoculant, they would often include non-fungi, or in this case, a fungi that is not a mycorrhizal fungi, trichoderma. And this trichoderma is a fungus that is basically a general symbiont or an epibiont, something that grows on the surface of plants that can kind of outcompete other things. So in all cases, actually, the Bacillus and the Pseudomonas here, these two groups in most cases are actually considered to be pathogenic groups. So if you know any pathogenic micro, you've probably seen these two genus names pop up because they're very prevalent. They're very prevalent in soil, but they're also very prevalent in human pathogens. But in all cases, and trichoderma also has pathogenic species, These are groups that are really just broad generalists that can help take space. They can grow, and they can hopefully outcompete other potential pathogens. And you'll see this a lot, and you'll see this a lot when you look at probiotics for human health as well, that a lot of the things that we eat in probiotics are just things that are taking up space. If you look at most probiotics, I'll let you shout to the other people in the room if you know what one of the main genera that you see on those probiotics is. What is one genus of probiotic that you always see in those? Now that you've yelled at people, hopefully. Lactobacillus is one that you see all the time. And lactobacillus is a really well-known human probiotic, both for food and for these probiotic pills that have been developed over the last couple decades. And lactobacillus is a member of the human gut microbiome, but it is not a very prevalent member. It's there. It is a small member of the community. It may be very important, but it also is not a big driver of that community function. It's something that is a totally safe member of your community to have. So it's something that is good there to have there just to keep other things out. And that's kind of similar to these two bacteria, I think, in these soil additives. So that leads me to my fun little Getty picture here. I really wanted to find a nice picture of a kid eating yogurt. It's yogurt. It's all yogurt. And so I do feel that a lot of these probiotics that we eat as humans and these plant inoculants are really at the level of yogurt. You're just giving them something that supports their plant health but doesn't necessarily promote plant health or plant growth. It doesn't give you maybe the big gains that you want to get out of it. Or maybe it does in the case of a small number of plants. And so my real question then is, how do we move past yogurt? How do we, both for human health, I think I'm very interested in that as well, even though I don't study human health. How do we move beyond everybody taking the same probiotic, every plant getting the same mycorrhizal fungal supplement, even if it does spell extreme with an X? I mean, that's hard to argue with. And the first thing I had on my list, how do we improve this, is study more plants. And so I did some quick searching when I was making this presentation, where I just did Google Scholar. If you know Google Scholar, it's a nice repository for nearly all scientific papers, and it's very easy to search. I used the common names for all of these organisms. I could have used the genus names, but I don't know half of these genus names. And it was an easy standard to use for all of them. And I just punched in a few of the most common things that I could think of. And I was surprised, actually, that wheat rhizosphere, so rhizosphere, that's your soil microbial region, the wheat rhizosphere, for some reason, has the most publications. That actually surprised me. I thought corn or pea might be the highest. Potato rhizosphere was pretty high. Soybean, 77,000. Pea. And then the first one that some of you on here might not know is Arabidopsis. So Arabidopsis is the plant for research. It is one of the canonical plants for doing plant research. And it's just a weed. It's just a fast-growing little weed. And it's very much equivalent to E. coli. E. coli is maybe not the most normal bacteria, but it's very easy to grow, and it grows really fast. And so that's why we study it. And Arabidopsis is kind of the same thing for plants. There are some plant people in here who could tell you the common name for Arabidopsis. Beets, I don't know why I put beets in here. Maybe I just wanted a purple word. Beets are 23,000. But then asparagus I put in here because I like asparagus. Tastes really good. There's only 4,600. So you see the difference between, obviously, I think you would predict this, the difference between soybean and asparagus. And then it's even worse if you go to something that isn't a food crop. Blue stem, generically, I chose blue stem, but big blue stem and little blue stem are a couple of common prairie plants. And there are prairie plants that are in our study group. And you see the number of those is much smaller. And I think a lot of these studies, too, are community studies. These are not direct studies on the blue stem rhizosphere. Whereas in the case of potato and corn, most of these studies are on these species specifically. So you see a huge difference, and this is just a small number of plants that I searched, and my plant knowledge is poor at best, and you can see a really wide variation in these. And so that's a problem. And when we get into natural ecosystems where we have mostly things that are not food crops, it's an even bigger problem. We don't know anything about those plants. And in fact, even most of these, we don't know all that much about what the microbes in their roots are doing in relation to those plants. Another area that I am very interested in is learning directly how plants and microbes interact. So that fuzzy figure that I showed earlier was kind of a joke at the expense of the lack of knowledge in this area. And that's really a challenging problem because you have a really diverse microbial community in contact with a plant that you maybe don't know that much about. So these plants like corn and potato and things like that, we do know a little bit about what exudates their roots pump out, what chemicals their roots produce to attract microbes, but we don't even know a full spectrum of that for most of those plants. So when you look in natural communities, it's even more of a challenge. And so I've been really fortunate to become involved with this lab at Berkeley National Labs through Trent Northern. I'll show him and his group later, but they've developed these little ecofabs. These are little plastic cells where you can grow a plant and keep its roots isolated. And these systems are very good for keeping the roots sterile if you want sterile roots, or inoculating the roots with a very specific microbial population. And you can insert probes and measure any sort of things that you want, or you can take samples out. And one of the things that they commonly do is they take samples away and do this little step here, analyze exudates, or mass spectrometry over here, where you can see exactly what's happening. So you can grow this teeny little plant. I have no idea what this plant is. I'm sure somebody in here does. You can grow these little plants, measure what chemicals their roots are putting out, and then see what the microbial population does to that chemical composition. Or even in their case, they would make an artificial exudate blend and feed that to microbes and see how those microbes interact with those chemicals. One caveat to this, of course, is that these are small plants, and very often these are just shoots. These are just young plants. And plants, we know plants change their exudate blend. They change their root chemistry as they grow. And so that's something that we're looking forward to, working on more. But a lot of this is really foundational work in studying these plants that have never been looked at before. Everybody has looked at the above ground traits, see what type of leaves they have, what their biomass is, and all of those sorts of things. And they haven't really focused too much on what's going on underneath because it is a lot harder to look at. But this system, we saw this as a very good system to start to get at this problem. And then the other thing, the last on my list of things to do is think beyond individuals. Think beyond each plant interacting with its microbial community, because we know that's not how the world works, right? We as scientists are very guilty of analyzing things in isolation. In microbiology especially, everything we want to look at is a single microbial population, a pure culture where we understand exactly what that microbe does. But in the natural world, of course, most microbes are interacting with a huge number of other things. We humans are interacting with a huge number of animals and plants and insects and microbes in our daily life. And that changes how we behave. So looking at these things in isolation may be a bit of a problem. And so the two pictures on the left here are from Nachusa. This is from a former undergraduate at NIU, Atlee Hargis, who just contacted me the other day, actually. After this announcement was made, he contacted me. And so he's a great photographer, and he took some really good pictures at Nachusa. And then the right side of this is at Morton Arboretum. And Morton Arboretum has a very controlled system that I'll talk about it later in this talk. So for those of you who haven't been or don't know much about it, the Nachusa grasslands is the first place that I really cut my teeth on doing soil research. And when I first moved to NIU, Nick Barber was here. Nick Barber was a faculty member that started the year before me, and he studied primarily insect-plant interactions. and he was trying to talk me into working at Nachusa and doing some soil microbiology, and I kind of told him he was crazy at the time because soil was terrifying. And this was in 2012. I really didn't think that we had the technology at that time to analyze microbes in soil because they are so diverse. It's kind of like taking an entire zoo full of animals and trying to figure out what's happening in that zoo. What's happening with all the animals? You know, it's a single question for thousands of different things that are happening at once. And so he did eventually talk me into it. It took him about a year. My first time I went out to Nachusa was in the fall of 2013. And the thing that convinced me is that Nachusa has a really well-designed, well-controlled system that would allow us to look at some of these questions without, some of the worries that we had. And so some of the worries that I had. And one of the reasons for that is they have what's called a chronosequence. They have a bunch of fields that have been planted at different time points. And their earliest field was planted in 1987. That's over here. It's called the main unit. And then at the time I started this project, the newest field was planted in 2014. 2012, I think, was the newest, the first year I started. And they also have a couple remnants, and these are sites that were never plowed. They were never planted. And so these are something of a reference, although these are mostly kind of weird. And so we went out there and did a lot of analyses on this soil to see exactly what was happening. And one of the first things that we saw is not new information. We saw something that many people have seen before when they look at natural communities. And that's that the older a site is, the longer this prairie has been planted as a prairie, the lower the relative proportion of nitrogen is. And that's represented by the nitrogen going down in this old sample. So this is more carbon, less nitrogen. This is more nitrogen, less carbon. And that's because we have these big, ridiculous molecules. So these are lignin, cellulose, things that plants produce. So lignin is a common woody plant fiber. Cellulose, all plants have. And these are they're just very hard to eat. These are really big, chunky molecules. Honestly, lignin is as big as some microbes. It is a huge molecule. And so we have these really big molecules that microbes can't interact with. A lot of these are metabolized by fungi only. So maybe fungi are the only things that degrade some of these. And so they stick around. So as plants grow and die and grow and die and grow and die through the seasons, you accumulate a lot of these things with relatively little nitrogen in them. And that nitrogen gets used up too, because as I said, that nitrogen, available nitrogen, is a limiting reagent. Life needs nitrogen, it needs a lot of it, and it kind of runs out. We also saw some of the other ones that I mentioned. The phosphorus runs out, and that nitrate, this is representative of the available nitrogen that's been converted. That cornfield had tons of nitrate in it. And then we saw the older a field got, the older ones are on the left, the lower that nitrogen number went. And the phosphorus stuck around a little bit longer. Phosphorus has some solids that are not water soluble that stick around in the soil, but it does go away over time. You have a general decline. So these fields that had been fertilized for years at Nachusa, they would take these cornfields and convert them into prairie fields. They have this history of the leftover fertilizer that takes a little while to go away. And in the summer of 2014, we had, as I mentioned, these couple summer fellowship students that came out, and they had a lot of time. Nick and I didn't want to do this on our own, so we forced a couple poor innocent undergrads to do. And they went out there every 2 weeks, and they took a lot of samples and sampled water, soil moisture, pH, and took samples for genetic sequencing. And that's, as I mentioned, Jared and Renee that were in the earlier picture. They were out there all summer sampling these. And that led to a really cool publication where we saw some really clear trends. And this figure, many of you probably won't have any idea what NMDS is, but NMDS figures are a way of taking this huge amount of data, huge amount of community data, and boiling it down to just the two-dimensional relationship between sites. And so each one of these dots here is a sample that they took. And keep in mind, they went the entire summer. They sampled, I think, 10 total times. Wait, that's 8 times over the summer. And they sampled 7 sites. They sampled some of our main sites because we couldn't have them do all of them all the time. We didn't have the money to sequence all of them, and they didn't have the time to do all these analyses on every site. So we did a small subset, and they saw that if you take each one of those samples, we see that the dots that represent each site, if they're closer to one another, that shows that those 2 sites have very similar microbial communities. So this red dot number 2 here and this burgundy dot number 7, these 2 have maybe nearly identical microbial communities because they're right on top of each other. And that's a 27-year-old prairie. That's the oldest planted prairie, the one planted in 1987. And then the remnant, the field that had never been plowed. So this looks like those old prairies, and then the next oldest one in pink here is 13 years old. They're really starting to look a lot like these remnants that had never been planted. And that was really cool. This kind of confirmed what we thought we would see. The young prairies, the 1, 2, and 3-year-old prairie, are over here on the other side, and they're kind of all over the place. They're really variable, and that also kind of supported what we saw. These fields change rapidly year to year. The 1-year-old prairie looks very different in 2 years and very different in 3 years. They change really quickly as the new plants, the prairie plants, take hold. One of the things we didn't expect to see is our agricultural field. This is a cornfield, lay mostly right in the middle of these 2. There's a weird outlier here. We expected them all to be out here, not related to anything. They kind of were in the middle, and we're still chewing on this. We still don't quite know what to make of it, but it looks like this cornfield really is kind of in some sort of waiting period. It's in a holding pattern where it's getting so much fertilizer that there's tons of nutrients, but there's often very little water because this soil doesn't hold water. If you've been in a cornfield, it's like being on a beach. That water has no organics in it. And so it could be that this is just a bunch of microbes in waiting. And when you give them a lot of nutrients, they go in one way, and then when that changes over time, they go in the other direction. And we see some of this successional change. We see this change over time, meaning the microbial community changes over time. And I won't go too much on this figure, partly because I'm talking for a long time here. But we do see that there are certain microbial communities that are really associated with young prairies. That's these ones on the left. And then other groups that are really strongly associated with old prairies. And these are consistent with a lot of other studies in Colorado and other parts of the world, really showing that some of these microbial groups that we don't know all that much about are really critical to soil function in prairies. And I can't go a single presentation without showing everybody how fun the raw data is to look at. And so I'm showing you this here mostly for shock value. I don't expect anybody to make much out of this. But this is kind of what our data looks like when we originally pull it out. Each of these samples, each bar is a sample here, and it has a distribution of different microbes in it. This blue, big light blue bar here is actinobacteria. It's a very common soil bacterium. And then there's some, like the gray one up here. If you can squint your eyes, you see this gray is really prevalent over here in this remnant site, but there's not much of it over here. These are the things that are differentiated. This gray one over here is, in fact, this Verruca microbius Spartobacterium, this really important one that's associated with old prairies. So you can kind of see that by looking at the raw data. This is something that we always look at the beginning to see what the data might look like if we just kind of guess at things. And this figure I put up here too because it's also showing a major artifact that we found with our data and a major problem that we've had to correct since, where we have this actinobacteria really high in 2013 and 2014 in our first couple of years doing this, and then it dropped. And that doesn't happen. There's no expectation that that's going to happen at every site. And so we've discovered that this is a problem in our sequencing, our processing pipeline. So you identify things that are mistakes, and it's good to find those things so that you can correct them. And we've also had students that have looked out there. This is Carly, a grad student of mine who did a lot of work in Echusa, and in particular, she was looking at how bison are changing the landscape. So our studies began before bison were introduced in Echusa, and during my time here, just before Carly's project, bison were introduced to Echusa. And so she was really interested in seeing how bison change this landscape. And so this is kind of a before landscape, right? Where we have our remnants. As I said, our remnants are different from our older planting here in green and our younger planting in blue. And this was her sequencing that she did before the study. These all look very different from one another. These are 3 different time periods and they look quite different. And she spent a summer going out there nearly daily, where she would take these fecal patties, bison poop, and monitor the soil around the bison poop to see how this poop impacted the soil. This feces has a lot of nutrients in it, a lot of available nutrients that soil microbes like, that plants like, and so she wanted to see how that was affecting it. And in fact, she saw some fairly clear changes where, as the feces was incubated for a long time, these are the dark filled samples. In the young prairie, the 2010 planting, and the remnant prairie, the very old planting, they started to move toward one another. It looked like this feces was making these 2 prairies look more similar to one another, which was not at all expected. And in fact, when she went out there the next year, those 3 prairies that looked completely different year 1 now look no difference at all, right? They're overlapping. And so there's some evidence here, and we're still working on this project, some evidence that these bison are drastically changing this landscape by changing the microbial communities. And that change is likely going to translate up to the plant communities. And it also probably has happened in the other direction, that the plant communities will affect the microbes that are in that system. And so, in talking about these landscapes, as I said, one of the things we want to do is understand these complex communities, but maybe we can't do it if we're looking at Nachusa, where there are hundreds of plants in every square meter. But there's an amazing study at Morton Arboretum started by Andrew Hipp on the right, and Megan Midgley has started working on this as well, that is a incredibly controlled sample study. And I'm not going to go over any of this because it's very complicated, but there are 437 plots. 437 is right there, the last one. And these plots have been laid out in such a way that they have a rich diversity of plant communities. And Andrew's interest, he's a plant person, his interest is on phylogenetic diversity and the interactions of plants. And so what you see on this figure that is hard to make out on here is we have black, which are the empty sites. These are the unimportant ones. These are the ones that were not planted. Any site that has a little word on the bottom of it, and you can see probably, even if you can't read it, you can see there are little words on the bottom of a lot of these. Those are monocultures. So a big portion of these plots are actually monocultures where a single plant has been planted all by itself in this 2 meter by 2 meter plot. And those monocultures are, there are 130 plants for those monocultures. But then there are also the other ones that you can't see as well that don't have words on the bottom are mixed plots. And those are plots where they've mixed plant species from this selection of these. And just a really little peek at it is this. So this is just a little bit of the spreadsheet to show each plant. So the plant shortened name is on the left here, and these are the plots that plant is in. And anytime you see a bold number, those are the monocultures. So each plant is present in 2 monoculture plots. I should get my arrow out here. Each plant is present in 2 monoculture plots. So there's 294, 298, and 51 for this plant. And then each plant is also present in some number of plots. And so some of them are not in as many. This one is only in 4 sites, whereas this one, some of you might recognize the name, is in quite a few. And this is the only one on here that I put to memory. This is Andropogon Gerardii. This is big blue stem. So this is the one I mentioned earlier when I mentioned the blue stem. This is a major prairie plant. They grow very large, and they are in our focal group, and so that's why it's highlighted in this sheet. So we're focusing on a few of these to start our study, and that big blue stem is one of them. And if you look long enough at this and you're crazy enough, you might be able to pick out 2 of these that share a site. So surely some of these might be present in 2 different sites, and so that means these 2 species are sharing a site together, and we can figure that out. And there are, as I said, 130 of these. It's a huge number. And the fact that we have these monocultures is what excited me the most. And the reason I hopped on this project, so this is Andrew's NSF project that he started a few years before our new one. And I saw these monocultures as an amazing way to say, hey, this here, let's focus on our andropogon up here. This here, this 272, the soil in that plot must look exactly like what Big Blue Stem wants for its microbes. Hopefully, because it's all in isolation, that soil is what that plant dreams of in terms of the microbes around it, with no other plant influencing it, which could be detrimental. And that's part of what Andrew's studying in his, is whether there are benefits or detriments to sharing a space with another plant. So this is a great way for us to get at exactly what this plant wants. And then if we take this andropogon and we look at it in combination in plot 234, how does that look compared to 272, right? Is 234 drastically different? Does 234 have any sort of relationship to 272? Can we see why it's different or how it's changed based on the other plants around it? And that was incredibly exciting to me. And that's a way to really start to pick apart the data. And so, of course, as in all things science, and this is just a teeny little basic science lesson, you always want context. And so one of the things that we did is we were out there sampling before these were planted. So we were out there a few weeks before the sites were planted. They had been cleared. They had been herbicided, and they had been allowed to settle for a little bit. And what we saw is what we hoped to see. And we did a subset. This is just 50 of the sites. And what we saw is that most of them look quite similar to one another. And you see they're not all identical. The negative control over here is nothing. This is basically garbage, and that's why it looks that way. So that's our negative control. There is one site here, this 373, that's a little funny. Maybe 400 is a little funny, but most of them look the same. We have a lot of that actinobacteria that I mentioned. That's just in soil, no matter what. And we have a lot of proteobacteria. These are also in soil. Proteobacteria is the group that has things like E. coli and Pseudomonas and all of those. And then we have this, my favorite category, other stuff. These are things that we don't know that much about. This green bar is uncharacterized groups. And that, you see, that's a big chunk. And so this is our starting point. So when we start looking at these communities and we're just starting to get this data available and start to put this together, because it's a huge undertaking with 437 sites, we're going to be able to see how those compare to where the soil started. And then, of course, as I mentioned, we have this collaboration, and this collaboration I'm super excited about with Trent and his postdoc Kate at Lawrence Berkeley to look at these ecofabs, these little systems. I've known Trent for 18 years. I went to grad school with him, so it's really good to be working with him again. And these cells are going to allow us to take each of those, hopefully, each of those 130 prairie plants and grow them in these little cells and understand exactly what chemicals they pump out of their roots and exactly what microbial interactions we might have. And then the last collaboration that I mentioned at the beginning that I haven't talked about yet is putting this all together, the machine learning. And I picked a figure here intentionally, knowing that Josh would probably hate it because I kind of hate it. And when you search for machine learning, you'll see hundreds of images that look like this. They're very generic, they're very stupid, and they're very much based off of this idea. Either you'll see a human or you'll see a brain. And that's because machine learning is a rebranding. Machine learning is what we used to call artificial intelligence. And artificial intelligence, of course, has a lot going along with it. It's kind of a loaded term. But what it really is just machine technology, computer technology that can mimic some of the things that human brains do. And one of the ways I usually explain this when I'm talking with my lab about it or talking with classes about it is a lot of the techniques in machine learning are similar to the classification that we do as humans. When you see a big animal in a forest with a kind of short nose, pointy ears, and a long tail, very lean, you very likely immediately know that that's a cat. But mathematically or scientifically, there are reasons that you know that's a cat. You have associated that with a cat because you've seen lions and you've seen house cats and you've seen dogs and other large mammals that don't look like that. This is why we are confused when we see something like a hyena, which we think is a dog, and a hyena is not a dog. It's actually closer related to cats. But it has features of a dog, right? And machine learning is kind of getting at some of these things that don't fit well within normal characterization. So if you only grew up in a city and the only things that you ever saw your entire life in terms of cats and dogs were house cats and a variety of dogs that people had as pets, if you then saw a lion, you might characterize that as a dog because it's the size or bigger of a dog, and cats are only this big, right? You've only seen cats that are this big. But that's because your data set was very limited. And so one of the really exciting things about this project is that we're expanding our data set. So the data set that we're putting together for this is going to expand beyond the house cat understanding of the few plants that we've characterized really well to the full community, the full global understanding of all of these prairie plants, this wide phylogenetic variety of plants, that we can get at their plant root characteristics and how they interact with microbes. And then ultimately, to do something that's probably even more annoying for Josh, is to glue it all together, to tie it all together. And this is really what computation means to biology across the board. Almost all aspects of biology require data analysis, and sequencing is certainly the most extreme of those. And so when we're looking at all of this data and we're trying to correlate the microbial interactions at Morton Arboretum in a very controlled system, we're hoping that we can use computational techniques to predict how those interactions might occur in a complicated site like Nachusa. And that's not an easy proposition. We're taking sites at Morton that have a dozen plants in each site and going out to Morton where we have hundreds in close proximity. We have plants at Nachusa that are not in Morton, but we have relatives of them. And so we're hoping that we can see, are plants that are related to each other similar in how their microbiomes interact? Can we replace them? Can we replace a plant that isn't present at Morton but is present in Nachusa with a close relative and things like that? And so it's really, it's hard to understate the importance of this machine learning and this computational analysis to tying our entire project together. But with that, I've gone on way longer than I said I would, so we'll definitely end and ask everybody if you have any questions, and thank you for listening. And our organizers were harassing me for not having the arrow up most times. I kept forgetting to bring the arrow up. I think you're still muted, Judy.
Can you hear me now?
Yeah.
Yes?
Yep.
Okay. Sorry about that.
If you have any questions, feel free to type them in the chat, type them in the QA.
Yes, so please do write in some questions or comments. And we know that there are a lot of you that are experts out there that have some comments about this. And it also gives you an opportunity in the next couple of minutes to refresh your drinks. Thank you, Dr. Swingley, for your presentation. I'm just going to take a couple of minutes to tell you, while you're writing in some questions, about some upcoming events that we're quite excited about. Our next STEM Cafe will also be virtual, and it's scheduled for October 7th. And this is important to tell you because STEM Fest actually this year is going to be starting October 1st, and it will be the entire month of October, culminating with an exciting day on October 31st when we'll have 4 virtual rooms and a stage where Jeremy will perform. So each Wednesday during the month of October, we are going to have a cafe on Wednesday. So they will be at 6 o'clock. And our first one is going to be Not So Creepy Critters, Exploring Reptiles and Amphibians with Dr. John Vanek. Some of you may have heard John Vanek before. He's a wildlife biologist and a postdoctoral fellow at NIU. And he's very engaging. And I think this is going to be a neat cafe because I think it's sort of a family type of thing that people can be really intrigued with. And so our second cafe will be about materials that are becoming scarce, and we need to figure out how to get them for a lot of our technology. It's quite going to be quite an intriguing talk. And then the third Wednesday will be one, or my colleague will be doing a future telling where she will be bringing in an author, a fiction author, and pairing that with some science experts and talking about the possibility of first contact with aliens, possibly. And so I can see that Dr. Swingley is already perking up because this is one of his favorite subjects. But anyway, that should be a lot of fun. And then the final one on Wednesday in October will be the science behind haunted houses. So we have a couple of experts that will be in on that, a technician and our dean of at the University for Visual Arts. And so that will be very exciting. So we have a whole month full. On Mondays, we're going to have Maker Mondays for kids. And on Saturdays, we're going to have workshops. And then we're going to get together on the 31st, where we'll have 4 virtual rooms going with lots of different choices. So anyway, if you want to know specifics, by the end of next week probably, or at least by October 1st, we should have the whole schedule on the website. But in the meantime, you can go into NIU's website and just put in STEMFest, and you will find out things that are being planned. And so you may want to get in there and search around. And also my colleague Jillian King Cargile has asked me to share with you about her event, which is going to be September 23rd from 6:30 to 8 p.m. And she is going to have Math and Science Are My Superpowers. It's a webinar that is free, September 23rd, 6:30 p.m. to 8. She's going to have an author, a physicist, and our Dean of Engineering who has a long career in biomechanical engineering. The trio will geek out about everything from swordplay to solid-state physics as they explore the connections between science fiction and science future. So again, if you go onto NIU STEAM, on NIU's website, you will find everything there. STEMFest, Future Telling, our upcoming science cafes, and so please do visit our website. Do you have any questions or comments? I guess we have a couple of them. One comment a little bit earlier by Aline. She said that actually the blue stem is one of her favorite landscaping grasses that you had mentioned earlier. And you have some compliments here.
Yeah, Aline mentioned she's a master gardener, and my wife also is. She does all the plant stuff. I'm not a planty person at all. I know next to nothing about them. I hope through this project to learn about a lot more of them, and certainly learn maybe some of the features that most people don't know about those same plants, learning about the underground characteristics.
We will give people just a couple more minutes to...
Yeah, I was telling Aline I know people in here, so they have no excuse not to harass me or ask me questions. But if not... in honor of the normal STEM Cafe tradition, I'm going to go get myself a drink after the talk is done. So.
And you might want to hold up your STEM glass, which you'll get another one for having presented here. And yeah, how cool is that?
Happened today to be using this glass. It's a good one.
So he's going for a whole set. And I know that some of you that have attended our STEM cafes in person are actually acquiring a set. So that's one of our favorite things to give out.
They're very nice glasses. I really like the etched glass.
They are.
Are people able to get those when you're doing these things virtually?
You mean like you?
No,
I
like
will.
anybody. There's no
Oh,
way for
well,
people to buy
we
them.
haven't been having drawings, virtual drawings, but I'm certain that I can come to campus to make sure you get one. But soon we hope to be back in our restaurants. We just all have to be good in the meantime and wear our masks and social distance. And we hope to be back to our restaurants. And I think our audience is asking us to actually do both. They're asking for live streaming and some virtual so they can get through all of them. So it'll be interesting what we can come up with after we get back to the restaurants.
Yeah, for sure.
We have some great partners.
Yeah, we have a question about presenting this information to middle school students. And there's a great quote that I remember years ago from Neil Gaiman. Some of you may know Neil Gaiman is an author who's written books for both adults and children. And somebody asked him about the difference between writing something for uh, children and the difference. And he said, well, when I try to think of an audience that's going to really analyze and dig into depth about my writing, that's writing for children. When you write for adults, it's just any old crap. And it's really true. I think when I've spoken with high schoolers about my research. I spoke last year or earlier this year. That wasn't last year, was it? At STEMfest. Feels like forever ago. To some grade schoolers.
Talked at STEM Fest last year.
Was it last year?
Yes.
Time has disappeared. And the impressive thing is that the younger students really pick things apart faster than most adults in my experience. And so presenting to them, very often presenting some of the anecdotes about scale and size, I found very valuable to get people to understand that scale of microbes versus trees and versus humans. And then getting students engaged in the activity. There are a lot of ways now for students to take samples themselves and maybe get sequence data or get microbial data or look under a microscope. And I find that most students love to have that hands-on experience for doing these analyses. But yeah, beyond that, I don't know. Microbiology is always a challenge. I always kind of admit my weakness in terms of trying to make things accessible. At the same level, I'm working on projects where I'm working in prairies where we have bison and we have plants and all of these really charismatic things that people just get. They just understand them innately. And so a lot of times I associate the concepts in microbiology with these real world terms, and it helps out in visualizing those and bringing those into perspective. Another question was, how do the monocultures at Morton attract or select for the microbes they prefer? And that's a really good question. And I think a lot of what we go into this thinking is that soil, even though we did a really broad sequence and we see they all kind of look the same, within that chart that I showed is a huge amount of diversity. And there's also a lot of rare species in there. So you're seeing the really big ones. You're seeing the actinobacteria and the proteobacteria. But even within those, there are some microbes. There may only be one or two of that microbe in the sample that I took. And when a plant is growing in that soil and pumping out certain chemicals, certain exudates, those may be very strongly selective to certain microbial groups. And so the expectation is that community that started before the planting, that started from whatever, just sort of wild grass, wild growth that was there, is going to change drastically as plants start to heavily influence that by dumping their exudates into the soil and continually enriching for the organisms that like those chemicals. The other thing that a lot of people have talked about in terms of this concept of specific microbial populations is an old term that was coined a long time ago, and people debate all the time whether it's true or not, is everything is everywhere and the environment selects, meaning any gram of soil has every potential for the bacteria in that sample. If you change it, if you heat it up, you'll get things that like the heat. If you make it wet, you'll get the things that like the water. If you make it salty, on and on. People have debated that a lot, but I think for the most part it seems to be true. Microbes are also very easily aerosolized. They get into the air very easily. And so at a place like Morton or at a place like Nachusa, the expectation is that microbes are being imported all the time. You're getting in foreign microbes all the time. If those foreign microbes come into a situation that they prefer, that is selecting for them the right nutrients or the right water or whatever it may be, then they have the potential to take off. And so I'm really very interested, since we have pairs of these monocultures, to see whether they have the same microbiome. The hope is that after a few years they will have the same microbiome, but that may not be true. Maybe they're wildly different, and that'll be a result as well. Thanks for the questions.
So we have a wise guy in the audience that is saying the stemware is very glassy.
Stemware, I didn't even think of that. Very nice.
While we're waiting for somebody to ask us a question, I do have the name of the talk for October 14th. It's The Secret Lives of Rare Elements, Finding Critical Materials for Green Energy. And so...
Who's giving that talk?
Mark Frank.
Mark Frank.
You know Mark Frank.
That should be a good talk.
He's talking about some of the materials that exist. Some of these materials exist in incredibly small amounts within the earth, and talked about, as we develop low-carbon, green energy sources, humans will rely more and more on these rare elements. Some of them that were identified earlier, but we didn't find a use for them before.
Well,
Anybody else have any?
Showing up and for listening quietly. Nobody interrupted me. And hopefully you'll join on some of these other ones. And thank you, Judy, for hosting.
Well, thank you very much, Dr. Swingley. I don't think anybody has any other comments. That doesn't mean your talk wasn't fantastic. I think they just understood maybe a lot of what you said, or maybe the opposite. Maybe there are a lot of questions here that need to be and that's exactly why you're doing the study, correct? That we need to find out how these different microbes react and how they are healthy for us. And maybe you want to cap or summarize your talk in just a minute or two, or a sentence or two.
A minute or two? No, we don't know what we don't know. That's my lifelong motto, is trying to figure out what we don't even know that we don't know. And that's true for my astrobiology work as well as this. And so they are very, very related. For a little while, since I got funded for this project, this soil project, I've thought, how do I name my lab? How do I come up with a lab theme? Because I'm studying soil and I'm studying exoplanets, planets around other stars. Those couldn't be further apart from one another. But they have that theme of, trying to dig into the deep interactions of how microbes and the environment interact with one another. So,
thank you, and I hope our audience will try to get the word out to the rest of the folks about our STEM Fest. I think we have a comment here. Thanks.
Yeah, just a very quick question about using my work in a timely manner. One of the greatest things of being involved with Nachusa grasslands is the land managers there are really interested in science and interested in using, science to inform what they do and how they plant new fields and how they manage those fields. And so a lot of the research that's been done at NIU, not my research, a lot of the other researchers that have been working in intrusive grasslands, the results that they get can be really rapidly turned into responses by NACHUSA and other land managers, and that's really cool. And microbiology has lagged behind that, and microbiology is so complex that a lot of this analysis has been harder to implement and harder to put into things. The follow-up from the same question was, how about farmers? And that's a whole different question. There's similar questions that a lot of people are looking at in terms of animal health and animal weight. One of the main ways that ranchers make their cattle heavier is by giving them trace doses of antibiotics, and that obviously has a lot of major concerns, but it's well known to increase the mass of those animals, and so from an economic standpoint, you can see why they do it. And similarly, from an economic standpoint, you can see why people fertilize your fields. And so if you find solutions, those solutions have to be equitable. They have to be as good at improving the crops or have some other reason to drive people to do that economically. Socially helps if you can make people think of the social implications of things that they're doing. That can help, but it's hard to beat the economics.
I think there was another question there.
Yeah, the other question, how do you factor for things that land in the area of the plants, decomposing organisms, etc.? Do they cause outliers? I think they do, and I think we'll always see outliers. And so when I showed that the baseline data for the Morton plots, all those 437 plots, there were a couple that were different. And those couple that were different may have been different soil types. There are differences in the land, even in that small scale. One side of it is very loaded with clay. The other side is more normal soil. But you can also, when you're sampling for microbiology, you can also just happen to get something. If you happen to get part of a root, if you happen to get part of an animal, if I happen to core through a worm, I may see results that look very different. And so what we try to do is just do enough data gathering, those things get washed out. Those things get lost in the background of all the rest of the data. But it's a good question. It certainly happens a lot in terms of microbial sampling.
Again, thank you, Dr. Wesley Swingley. It was a pleasure to have you this evening. Again, our next STEM Cafe is October 7th, Not So Creepy Critters, Exploring Reptiles and Amphibians. And October 14th, The Secret Lives of Rare Elements, Finding Critical Materials for Green Energy with Dr. Mark Frank, who is a professor of geology and environmental geosciences. I want to thank Aline Click and Raz Abdul Surani. We could not have done this without them. Aline is our NIU Director of Web Media and E-Learning, and they have assisted us tonight with Adobe Connect. We could not do it without them. This event has been recorded, and it will be available in about a week. If anyone is interested, or if you know anybody interested in sponsoring a STEM Cafe, please do go to NIU STEAM's website to find the link, or email me. It's https://go.niu.edu/give-NIU-STEAM. But you can find that on NIU STEAM's website, and so we would be grateful if you pass that word along. Thank you for joining us, and please provide your feedback by emailing jdymond@niu.edu. Thank you, and have a wonderful evening.
Thanks, everybody.